And then go back. And then put the laser.
It sounded like it's not connected.
He has to do something here.
Right. Finish this. And
Yeah. Yeah, I'm just gonna check if it's working.
and could you check the sound?
Okay, let's see if we can get this to go. Okay, sorry about that, folks. My meeting in the dean's office went a lot longer than I had anticipated that was going to go, and I couldn't actually sneak out on the dean, so just bear with me. Actually, Elaine is uploading the PowerPoint files. If you don't have a handout, it's going to be uploaded on Facebook first, and hopefully later on your PSL300 webpage for you. So if you go and check, you should be able to download the files in a few moments once Elaine gets it up there. And thanks, Elaine, for doing that. So I just wanted to go through a couple of things really quickly with you as an overview. I'm not going to be re-lecturing to you. I'm just going to be going through a couple of things that you should be thinking about. So all the questions are taken directly from the lecture notes. You don't have to worry about going and getting a textbook or anything else. Majority of the questions are going to be application type. There, you got to stop moving your foot because it's really distracting. So I submitted 26 questions in total. I'm not sure how many are actually going to be used. I know that you'll have about 26-ish questions apparently on the midterm, but I don't know of the 26 how many are actually going to be used. All of them are taken directly from your slides, from the lecture material. This review isn't comprehensive. It's just to make sure that you're going to be able to do well. If you're going to try to get perfect, you have to go through your notes, not just through the review questions that are here. So it's roughly about 6 to 7 questions per lecture. Your final exam, just to get you ready for that at the same time, because you should be studying for both, to be quite honest. When you're going and studying the final exam, I've been asked to supply 20 questions, which means that it's about one question per each hour of lecture I've given you. So lots of luck trying to study for that. You basically have to study everything for like one question from each hour. Not sure how fair that is for all of you, but that's what I was asked to do, and that's what I'm going to be trying to do. So I'm going to be going through a guide of how you should view and think about the slides as you start going through and studying. I want you to start thinking about it the way that I do when I'm trying to come up with these different questions, okay? So I told you that you should make sure that you go through and know how to identify different types of cutaneous receptors. And when you do so, I also want you to start thinking about which tracts these things are using. So some of them use spinal thalamic tracts. So it's not just enough for you to memorize that A alpha and A beta fibers are large diameter myelinated. I don't really care about the speed. I don't really care about you memorizing this. I want you to start putting some of this together. So is this going to be using the spinal thalamic tract? Is it going to be using the dorsal column medial lemniscus tract? And if you're thinking about them, then yes, these are going to be sending their axons up along the dorsal column, medial lemniscus, as well as spinal thalamic, and you should be thinking about A delta and C in exactly the same way. Please don't just sit there memorizing this slide. That's a good first step, but you need to start thinking about how is it going to be applied. So if he touches something over here and he's not giving me all the information and he's telling me that it's a myelinated fiber and it's going up the dorsal column, what's the target for the dorsal column that I might see an effect in? That's how you should be thinking about it, okay? Not just, I'm going to get perfect because I'm memorizing this slide. I think after the experience of your first term test, you'll know that I integrate information as much as possible. So try to sit there integrating a couple of these slides at a time. So in terms of this slide, this is also another slide that you should make sure that you're spending some time on. So many of you will know that I like molecular mechanisms, so you should be spending some time going over what sorts of signals lead to different types of pain responses. So are you activating the ENaC-DEG type? Are you activating TRPV1? How do you sensitize TRPV1, which is over here? And this is through like the sensitization, making it more painful, or making the receptor activate for longer periods of time, is through the activation of the a GPCR, either through ATP or bradykinin, that increases the activity of the TRPV1 receptor that is involved in pain sensation. Okay, and for TRPM8, we're not sure, but this is related to your feeling of cold, painful types of cold on free nerve endings. And again, put all of this in context. These are things that are found on free nerve endings. Would we expect to see this somewhere on a Pacinian corpuscle? No. So again, read through the questions carefully. There's always something embedded in the question that relates back to the basic mechanism, the basic action that I'm looking for, that you should be able to identify. So if I'm putting in a receptor, you should know that it's probably on a free nerve ending related to like the polymodal C fiber, or a pain fiber, what tracks it uses, where it's targeting in the brain. Start thinking along those lines, okay? Okay, so I am going to tell you that you should also make sure that you know about the reflexes, and the reflexes in particular. A lot of you were asking about this, and I'm hoping that you've at least gone through this muscle spindle reflex a little bit, and you should know some details, basic details that I won't be testing you on. Like, I'm not going to be asking you which of the following statements about the muscle spindle reflex is correct. It's monosynaptic. It involves a muscle spindle. It involves like alpha motor neurons. That's not the type of question I'm going to ask you, but I do need you to make sure that you know all the characteristic features of the muscle spindle reaction or reflex. And do you know all of the parts of the reflex? How it works? What is the receptor for this? For the actual muscle stretch, what's the receptor? So it's the intrafusal muscle fibers which make up the muscle spindle. That's what you're seeing here. And this intrafusal fiber is something that I didn't spend a lot of time on. And on the last lecture yesterday, we talked about all these extrafusal fibers, the things that actually make up muscle, the glycolytic, the oxidative, the mixed types. And again, one of the things that happens is in this stretch response, when you have this applied stretch, the first response back is that you get muscle contraction. And one of the things that happens is that this fires through the 1A afferent. Remember, proprioceptors don't use the A alpha or the A beta or the delta, A delta system. They use type 1A, type 1B, type 2. So this is a fast response back centrally, and it causes the muscle to contract, which is what you're seeing here. Unfortunately, though, that means that this muscle spindle is no longer sensitive, right? Physiologically, what does that represent? It's no longer sensitive. What does that actually mean? So I tried this yesterday. I'm hoping it still kind of works for you. But if this is sort of the muscle that you're looking at, and the muscle that you have, this thing in the middle, which detects any kind of tension or any pull. So when it pulls on it, this is the muscle spindle in the middle, right? And this is your muscle. And what we've done now is we've applied weight, a passive stretch, and it's done this. That part you're all clear on, right? So this sends a signal back to the spine saying that there is a weight on your arm, you should do something about it. So what happens is this muscle spindle and the extrafusal fibers, the muscle around it, actually does this, okay? And what can you tell me about the muscle spindle in the middle? Is it still going to be able to detect any more pull on it? It's not going to be able to do that. And so it has to be resensitized through the gamma motor neuron. So the muscle is contracted. If I put more weight on the muscle when it was like this, after it contracted, so this is the normal step. So it pulls, the muscle contracts, the muscle spindle can't recognize if more weight is going to be put on it. You would drop the weight, but you don't do that because every time I add more weight, the gamma motor neuron fires and it it contracts this muscle spindle, which is what is being shown to you here, that this inactivated muscle spindle has to be resensitized to the firing of this gamma motor neuron, okay, in the contracted state. And that's, again, the only thing that I want you to take away from this. Again, the other things that I need you to make sure that you know is that you should know the different types of axonal fibers, just as a sort of base, that you should make sure that you know all about the C fibers, the A delta. And I actually put A delta up here this way for a particular reason. If you see AD and it's not A delta on the test, you should just assume that it's A delta. Okay, so if you see AB and it's not A beta, for whatever reasons, and Dr. Key didn't have the symbol sign on, you should just make sure that it's the A beta. Same thing for A alpha. Okay, so unless there is something dramatically wrong with what's put up there, like an AF or something, then you should make no assumptions that it doesn't represent like the A delta fiber. So again, you should make sure that you know the whole pathway, in other words. I'm asking you to think beyond just what's on the slide. Think about the whole pathway, and do you know what activates these different nociceptors, which goes back to this slide? What activates them? What sorts of pain responses? Where it's going? Do you know about the sort of gate theory of pain and the gate control theory of pain? Do you know how pain, referred pain works? What system is it using? What do wide dynamic range neurons have to do with this? If that doesn't make any sense to you, what I just told you about the gate control theory, about referred pain, and about wide dynamic range neurons, then you should go back and review sort of the last few slides from last Tuesday's lecture. I know this slide is a little different than the one that you have, but from the slide that you have, when we're talking about the spinal thalamic tract, I'm going to ask you a question about the spinal thalamic tract. It is inevitable that you're going to get a spinal thalamic tract question, okay? At least one, by the way. So at least one question. You should know, for example, that there is a difference between the anterior and the lateral spinal thalamic tract. There are differences in where the neurons, when they are sending their signals in, at what level, like are they in lamina 1 or are they in lamina 5? Where do they send their axons up? Where do the next sets of neurons send their axons up? And what sorts of information are conveyed more in one type of tract versus another? And again, for me, that's an easy question. A really quick question. Yep. Yeah. So that's up in the brain. It's medial in the brain. But when we're talking about in the spinal cord, which is at the bottom, when we're talking about the anterior spinal thalamic tract, it takes a medial or a lateral tract up in the brain, which is what you're seeing at the top. On the bottom, it's referring to the spinal thalamic tract. Okay, so down here is where we're talking about the spinal thalamic tract, and when you get up into the brain, which is the upper part of the diagram that you have, then we're talking about whether it goes medially or laterally. And that part I'm not as concerned about. Okay. So again, you should make sure that you go over the mechanical gating, the control of pain. These are those wide dynamic range neurons that I showed you. Where do we find these wide dynamic range neurons? What tracts? Spinal thalamic for sure. Sorry? Spinal reticular thalamic. Do we find these in the dorsal columns medial lemniscus? We do not. And of the two, we only have the ones that go from the spine up into either the thalamus or the reticular formation before the thalamus. Should also make sure that you know things that I've talked about in class, like the substantia gelatinosa, right? So we've talked about that, and you should know what sorts of things are found in the substantia gelatinosa, where it's sort of located, and why these might be important in terms of the gate control theory of pain. Again, I told you not to know these. You don't have to worry about following up on any of this, just as I described it to you in lecture. So I'm going to ask you to make sure that you know the major features of the reflexes. So I was asked to come up with challenging questions. Not really sure what challenging questions actually means, but I also felt that just knowing the differences between the reflexes and making sure that you knew the key differences between the reflexes was a challenging but also fair question. So you should expect to get one question that I would think, and don't take this the wrong way, I think it's fairly simple, where you will be given some characteristic features of a reflex and then asked to comment on it in the answer parts at the bottom. And I think it's a straightforward, easy, but somewhat challenging one because you need to know all the reflexes that we talked about. Okay, so if you can know the major features of the reflexes, one or two of those questions will be really easy for you. So other things that you should be thinking about, what are the features of neurons that are found in the intermediate zone? So including things like the flexor burst and extensor burst generators. What other types of neurons do we find in the intermediate zone? What are some of the other features? Anything? Can you name me anything else that you find in the intermediate zone? Anything? Yes? So commissural interneurons, do we find them there? Do we find in the lumbar part of the spinal cord, do we find flexor burst generators in the intermediate zone? Yes, we do, right? So this is where they exist. They're not in the dorsal, they're not in the ventral. Like just general questions like that. Those are ones that I would consider challenging, where you have to integrate over many slides. As long as you know your material, I think it's straightforward, but you'd have to actually know your slides well. What is a BET cell? Because I just covered that yesterday, or the day before, actually. So can you tell me the difference, without looking it up in your notes, what the difference between a BET cell and a Renshaw cell is? Why does everyone look like they just ate something really sour? Like everyone's face is like, it's like the worst thing to see. Is the BET cell found in the brain or in the spinal cord? BET cell? Up in the brain, so it's part of the motor cortex. It's involved in actually initiating movement. It's one of those really large neurons that we find up there. And the Renshaw cell that I talked to you about yesterday is also related to motor neurons, but they're the inhibitory cells found in the ventral horn that stop the motor neurons from overfiring. So as you recruit and speed up the impulses. So again, knowing the different cell types goes a long way for you to be doing well. And do you know some examples of different transcortical reflexes where the reflex doesn't just stop in the spinal cord, it goes all the way up into the cortex and then comes back down? The major feature in a transcortical reflex is that they're slower, not by a lot, but they are slower. The grasp reflex, where you have slippage, that would be an example. The proprioceptive joint coordination would be an example of a transcortical reflex, and just making sure that you know that. Okay, some other things to, again, just think about, and I have to apologize, this title is incorrect. It should just be some general features. So when you're looking at the CNS and you're looking at regions of the intermediate zones and cerebellum, start thinking about what types of neurons, again, it's an integrative, challenging question, what types of neurons do you have tonic activity in? So I want you to start thinking about things that way, not just about purely memorizing it, but finding themes, finding things that you can have in common. Tonically activated neurons. So we know flexor burst generator, that's tonically active. Do you know any other tonically active types? Which other ones do you know of? So you know that in the cerebellum, your nuclei are tonically active, right? And you actually have to inhibit those. So is the globose type nuclei in the cerebellum tonically active? Yes, it is tonically active. So I want you to start thinking as you're going through your slides about some of these common themes. Like we talked about tonic activation of the mesencephalic locomotory region, that's tonically active. So are the flexor burst generators in the spinal cord, they're also tonically active. Make sure that you go through, because again, I like to, even though I'm asked to create challenging questions, I also like to have some questions that are relatively straightforward as long as you know the material. I told you that you should know the structure and the circuit in the cerebellum. So do you know where the granule cells are? Do you know what synapses come from granule cells to the Purkinje cells? What are they called? Like those simple types of questions, I like to have a couple of them on as well. And again, I look at them as sort of, as long as you've studied it, they're relatively straightforward marks. Examples of what the red nucleus does and what the rubrospinal tract from the red nucleus does. A lot of students kind of just gloss over it. They don't think it's going to be asked, and sure enough, you're going to get it. If it's not on the term test this time, you're going to get it on the final exam, because really, you're studying for both at the same time. Your final exam's not that far away, and you need to get as much studying done for that at the same time. I'm going to tell you a couple of things. So you don't have to memorize the diagrams related to the flexor, extensor, leg, heel strike, that diagram at the bottom where it's sort of split into two sections. But I do want you to know the overall features of what leg flexion looks like, what extension looks like, what sorts of receptors might be activated in like the extensor thrust, etc. And I want you to also make sure that you understand the mechanisms that I showed you here about the flexor burst generator and the reciprocal inhibition, which is what we're seeing here. In the intermediate zone, one of the core principles or concepts is that there is reciprocal inhibition, whether we're talking spinal reflexes or we're talking about reflexive locomotion in terms of central pattern generators, we have reciprocal inhibition. Can you give me an example in the spinal cord of reciprocal inhibition? Because maybe I'd ask you that on your test. Yeah?
The sleep-wake cycle?
In the spinal cord?
Oh,
So right off the top, no, not in the spinal cord. What other ones? So do you remember when we have the muscle spindle being stretched? We usually just talk about contraction. What has to happen on the other side? The extensor has to be activated or has to be inhibited? It has to be inhibited. So that's an example of where you might, through these intermediate zone interneurons, also show reciprocal inhibition. And that's where I'm asking you to make sure that you're thinking about it in terms of larger themes rather than just purely memorizing things. Again, I want you to make sure that you spend time going over this diagram. I'm going to make those pretty straightforward, to be quite honest. Usually I like the types of questions where you know, someone has developed a new way to stain or isolate different types of fiber tracks, or a new way to stain the cell bodies of different cell types. Where might this be found? Again, this is a really good example of this. I'll ask you to make sure that you know this particular diagram as well, because it's important. And knowing, for example, the details associated with fine skills through the dentate or the globose embelliform, reach and grasp. And again, don't forget that I actually told you that the interposed is the name for both nuclei. So if you see this and you see it for reach and grasp, you should also equate that as well for the embelliform and the globose. And again, just in terms of posture and locomotion, make sure that you're spending time going over all of the different structures that I told you. Again, all of these are tonically, spontaneously active. They're all on all the time. Again, something to maybe think about now in studying over the weekend, coming up with things that you can group together as themes in terms of what you're actually studying and what you're seeing. So in terms of this particular structure, I'm not going to be asking you about any of the things that I haven't covered. So I won't be asking you like, oh, okay, between the D1 neurons that are found up in the striatum and the globus pallidus internus, what's the neurotransmitters being released? I never told you about that, so you don't have to worry about the details there. But I do want you to know about things that are on this slide in terms of dopamine helping to facilitate this striatal pathway, palatal pathway, because the pallidum is there to cause disinhibition. And remember that sort of disinhibition pathway. So I do want you to make sure that you know, again, like which neurons are tonically active. Make sure you're aware of all of that. Make sure that you know how this pathway works in terms of disinhibition, and then you'll be okay. and hopefully you'll spend some time going over it. Again, I took a look at the notes. There are a lot of notes, but if you're spending time over the weekend going through it, reviewing it, and spending where you have to memorize things like on these slides here, and you're spending time memorizing it, I don't think it's that bad, to be quite honest. It's not as bad as Vision was in terms of the sheer number of things. There are some details that you need to know, but most of it is conceptual at this point. And there's some fine details that you have to fill in. Again, make sure that you go through this, because you get 6 questions. Sorry, you get 7 questions on this lecture that I gave you on the autonomic nervous system, some of the general features of the autonomic nervous system. Again, the ones that you'll get based on the general features of the autonomic nervous system, as long as you know these are myelinated, and they can be shorter or longer depending on sympathetic, parasympathetic. We always release acetylcholine from this group of synapses, and this group of synapses will either release acetylcholine or noradrenaline. Then you should be fine. You'll need to think about the question, but as long as you have those basic concepts down, you'll be good. The other basic concept is the sympathetic nervous system originates only in the spinal cord, so at the thoracic level all the way down to the lumbar, so it's thoracolumbar for the most part, and some of you were pointing out that some of these are dashed lines, so they're not myelinated. The majority of these are myelinated axons that you see, and again, we'll talk about this other structure over here, which is the parasympathetic nervous system, which is the cranial, and then the sacral. So make sure that you know the differences between the different origins of the sympathetic, parasympathetic, and the sympathetic system. Again, another reminder of using this as a diagram slide to make sure that you know that acetylcholine is being released here. I talked about it, so you should talk about it. What is the receptor that's found on the postganglionic axon? Always. It's always nicotinic, right? And it's always nicotinic here. It doesn't mean that it's always nicotinic on the parasympathetic postganglionic target, whatever it is. Doesn't always have to be nicotinic. It could be a muscarinic receptor, but you should know that as well. Like muscarinic, acetylcholine is being released at the parasympathetic, noradrenaline is being released at the sympathetic. Last thing I'll just make note of, I did talk about this as well, that when it comes out of the spinal cord, we usually talk about it coming out of the intermediate region on the lateral part of the spinal cord as it makes that preganglionic target over here. And again, a good sort of summary slide to make sure that you go through it. I'm not going to be asking you a specific question like just drawn up from this. I didn't cover all of this. It's more about giving you an overview of how these things work. Make sure that you understand that. And I think for the most part, unless I'm completely mistaken, it's pretty straightforward for you, right? And the 2 questions or so that I'm going to submit for that are pretty straightforward as well. So one thing I was a little worried about the other day is some of these autonomic nervous centers that are under the control of various neuromodulators like serotonin or norepinephrine. When I asked you guys in class, a lot of you weren't really sure. You should know like where the origin of things like the acetylcholinergic system, you should know about the reticular formation, the pontine reticular formation as being the major site for acetylcholine synthesis, and then they send their axonal projections and then they release at the other targets. Another smaller group, which is the basal nucleus of Meinhardt, I mentioned in passing, but this is the big one, the pontine reticular formation. That's where acetylcholine is formed. Even though we have acetylcholine throughout the brain, this is the major site. The raphe nuclei are the major site for the serotonin synthesis. So without those, you would have no serotonin at all in your brain. If you were to completely lesion this, all serotonin production in your brain would stop. You wouldn't have anything to be released. Again, here as well, the locus coeruleus is the major site of the synthesis of norepinephrine. So if we wiped out the locus coeruleus neurons that are found here in the the brainstem and midbrain region, you would completely eliminate the norepinephrine's system. So again, make sure that you're aware of that. Yep.
So for the last 3 slides,
Yep.
do we need to know just the nucleus responsible for each neurotransmitter, or anything else other than, or do we need to know also the functions of each neurotransmitter?
I think I mentioned in class one other thing about this, right? Which is what? Do you remember? Yeah, so the one other thing that I mentioned in class is that for the most part, this particular system, the acetylcholinergic system, sends its projections out into the cortex. It doesn't send it into the cerebellum. Some of them actually do send it into the cerebellum, and so it can modulate the cerebellum, but not all. Yes. Yes? For serotonin, no. No, I might give you the name of a drug. A drug is used to lesion off this nucleus. What might the effects be? But no. And again, you can see here, for example, the cerebellum is impacted by the norepinephrine system. So I think those are the only things that I asked you to make sure that you knew on the slides. Like anything else that's kind of buried in here. Um, like I didn't talk about the basal ganglia being a potential target. I didn't talk about the medial septal nuclei, so you don't have to worry about them. Okay, so again, make sure you're also spending time. This is sort of the overview slide that we had. You are going to get, as I mentioned in class, a couple of questions related to the different roles of the hypothalamus. You might as well learn these things now and review them as much as you can, because when you come back to the final exam, it's cumulative, and knowing as much of it as you can now will help you an awful lot on the final exam. So make sure that you go through things related to the reticular formation and temperature. That's an important one. Make sure that you, the suprachiasmatic nucleus one and the retina, you don't have to know. So this slide and this part of the slide, and then the diagram of the pathways and the sympathetic, parasympathetic, and the radial circular muscles, you don't have to know those. It's not on the test, it's not on the final exam. Okay, so don't worry about those. Don't worry about the retinal response. You only have to worry about, and not even this one here. It's this one, the cardiovascular center, that you need to make sure that you understand how it's sort of an inhibitory system, and how this rostral half is important and the caudal half is important. You also have to know this, and to be honest, I could ask a lot of questions about this, but I'm only asked to give you 7, and so it's likely if you don't see it this time, you're going to see a couple of questions related to it on um, the next round. And what you should do, if you can, is to go and sit and go through this slide here and know the nuclei in the hypothalamus. So these are the hypothalamic neurons. You should make sure that you go through the nuclei that you see here related to feeding behavior. Which ones turn them on, which ones turn them off, which ones cause stimulation, and which ones will inhibit different groups of neurons. And again, because things are kind of all over the place, some of these are mutually inhibitory. So again, maybe think about that. These are actually mutually inhibitory, reciprocal inhibition, if you want to think of it that way. So we're seeing another example in the hypothalamus. Maybe think about what sorts of stimuli on the next diagram actually work in a positive or negative manner. The traveling up the vagus, not so important, but just knowing ghrelin is a positive stimulator of feeding behavior, and all of these other ones are negative. So having too much leptin, it's like you've just eaten. If you have a lot of things that you've just eaten, you'll have more leptin, you'll have more insulin, you'll have more PYY, you'll have more CCK. Anything that looks like you've just eaten will turn off, in theory, the different brain structures related to feeding behavior, including things like glucose. And if you go through them, you'll see that all of these are related to having food in your system, right? So hopefully you'll have time to go through that. The only other thing I'm going to mention to you, when you go through the circadian rhythm section, I tried to keep the questions, at least on the term test, the ones I'm submitting, as straightforward as possible. So going through the slides, knowing about the different systems that are involved, including like slides related to melatonin, even though we didn't spend a lot of time on it. And this slide here is going to be really key. The only other thing I'm going to mention to you is I think your notes actually talk about this as orexin, right? In your notes, it's highlighted as orexin. But I use these terms interchangeably, and I think I mentioned it in class. So if you suddenly see hypocretin, I think I've mentioned it in class that it's the same as orexin. You should make sure that even if your notes are not like blazing with like hypocretin equals orexin, you should make sure that you're aware of this if I'm using them interchangeably. And again, you should know how they're working in a mutually inhibitory manner. So you might get a question. In fact, maybe I'll do that question. You might get a question related to which of these two pairs are inhibiting each other when the other one is active, right? And here's another example of where you get one active, the other one is inactive. Shall we give that question a shot? I just have it in my head right now, so shall we give it a shot? I think it'd be a pretty straightforward question. Oops, I don't know why this thing keeps going out on me. So just a couple of things for you when you're studying and when you're making notes. And if it's not for this term test, it's also for the final exam. Wherever possible, try to work out the entire pathways. Don't just learn it slide by slide, in other words. That doesn't really work well for you in this particular term test, nor does it work really well for you in the upcoming final exam. So wherever possible, because it might be you've gotten all this information about a stimulus on your skin, and then you don't know the pathway that it's activating, that's not very useful for you, right? Because that's the way that I like to write questions. Does it involve interneurons or not? If so, is it inhibitory or not? That reciprocal inhibition that we talked about. You don't need to know in your notes, and I want to be really clear on this, you don't need to know about the vestibulospinal, vestibular ocular reflexes, the ones that I told you you don't have to know. You definitely don't have to go back and worry that they're going to be on your term test or your final exam. The features of the CPGs, how they work within the lumbar spinal cord is important. I'm going to tell you that if you don't get it this time, and the question's not used this time, it's going to be used on your final exam. You should make sure that you go through the mesencephalic locomotory region slide, where you learn about how you actually initiate movement and the different tracks that are involved. Make sure you know that. I've also mentioned previously about the red nucleus and how they're different than the reticulospinal tracks. Make sure you go over that slide as well. Lots of tracks. Again, I'm re-emphasizing the transcortical reflexes and the different premotor areas, because I usually stick in a question about premotor areas. Do you know what premotor areas are involved in language, speech? Do you know the premotor areas that might be involved in the features of the premotor areas involved in grasping reflexes, etc.? So again, you should also go through how the basal ganglia work to control movement, the inhibition, disinhibition, the circuitry involved, and again, the cerebellum. Like, again, I think those are easy questions, so make sure that you do those. So these are the types of questions again, and there's some, and these are sort of the level of difficulty. I'm not trying to be obtuse about it. I believe in being as open about it as possible. These are, I think, Elaine, are the things uploaded for everyone? Yep. Okay. So I think you have it exactly as I've shown it to you. So you don't have to worry if you can't copy it down. It's all available on your Facebook group. And if someone can also upload it to the Blackboard portal page as well, that would be very useful. And I want you to think about these questions, because this is the way that I sit there and I come up with these questions. Based on what I've told you, you should actually be able to re-engineer or reverse engineer like my thought processes and how I came up with these questions, because this is the same way that I'm doing it for your questions right now. And again, you should make sure that you are familiar with all of these, and you should be comfortable with this. Normally, I'd actually give you sort of a review package where you have like more practice questions based on what we went through. So I don't know if you're a trustworthy group or not. Last year, the class swore up and down that they would not post this on like Student Buddy or whatever you guys are using now. And guess what? I found it on Student Buddy. So Normally, I'd like to give you some practice questions. You guys swear up and down that you're not going to be posting it on Study Buddy, Student Buddy, whatever it's called. You don't know that it existed? It's a great site. You should go for it. Yeah, I think it's a great site if you like paying money for things that are supposed to be free. Um, so in theory, I will somehow arrange it for it to be uploaded. Because if I upload it, Dr. Key might get upset. So if it gets uploaded by a student onto like the lecture recording links accidentally somehow, I'm not sure how, or on your Facebook study group and you promise that it's not going to be uploaded anywhere else, then I guess I'd be okay with it, because I know that that's one of the ways that you like to study, and some of these questions might be useful. And again, when you're, this is again, as I just told you, this is exactly the way I like asking questions, right? So there's a new staining technique, you're looking at axonal tracks, and this is actually, this was a really specific question for that particular year, but you should actually be able to figure out the answer, even though it sounds like it's something you might not know. You should be able to figure out this answer even without all the other details of it. So I'm going to wish you the best of luck on your exam on Tuesday. So two things that I'm going to ask you to do. Make sure that you are studying over the weekend. Don't leave it till Monday. It's not going to work well for you. If you need extra office hours, I'm in the office in the afternoon on Monday, and then again on Tuesday if you have like last minute questions or you just want to make sure that you understand it. And I'm going to ask someone in the class to upload the practice questions for you, and best of luck studying. Have a good weekend. Okay, and hopefully you guys will have a better idea of how it actually